Welcome everybody to the first of three many webinars on open science and comparative cognition research. This one is entitled Open Methods in Comparative Cognition Research. And we are thrilled to host this online event and to see both familiar and new faces joining us. I am Nicolas Alessandroni, and with me today is Laurent Prétot. Together we lead the Many One project, which is a large-scale study of reversal learning across taxa, and we'll be guiding you through today's session. This webinar series was created to give the comparative cognition community practical tools and training in open science, helping to bridge the gap between its conceptual foundations and everyday practice. And this open webinar series is made possible thanks to the generous support of the Open Research Community Accelerator, or GAP. We have the pleasure of welcoming today Emma Tecwyn and Ben Farrar, our two speakers, expert speakers. Emma Tecwyn is a senior lecturer at York St. John University in the UK. She has a background spanning biological sciences, animal behavior, and psychology, and is interested in the evolutionary and developmental origins of cognitive abilities, particularly those implicated in problem solving and decision making. She uses behavioral experiments to investigate a range of cognitive abilities in diverse groups, with past projects having examined planning in great apes, intuitive statistics in capuchin monkeys, understanding of gravity and solidity in dogs, causal reasoning in toddlers, and experience of time in children. Emma strives for open, transparent, and reproducible research practices in her work, but knows there is always more to learn. Ben Farrar is a senior consultant at Humanity, where he specializes in providing evidence on the comparative effectiveness of medicines to healthcare services. Ben has a PhD in comparative cognition, which focused on replication and research methods in the study of animal cognition. And with that being said, we will open the floor to Emma, who's going to be our first speaker. Just so everybody knows, we will have presentations of about 25 minutes each, and after that, we will open the floor for an open discussion where we hope everybody can contribute freely. So without further ado, Emma, the floor is all yours.
Thank you very much. Okay, how are we doing? Can someone give me a thumbs up if you can see the slides?
Perfect.
Brilliant, thanks, thank you. Okay, so thank you so much. I'm really excited to be here and involved in this discussion today. So I will dive right in. So I'm going to start by giving a little bit of context to the challenges that we in comparative cognition face, in particular in terms of sort of designing methods appropriate for a diverse species. And then I'm going to focus on three sort of key questions or prompts that we agreed that both Ben and I will sort of respond to and share our thoughts around. So the first, and the one that I'm going to spend the most time talking about, is what are the main challenges in ensuring comparability of methods across species, which is obviously super relevant to many. I will talk briefly about aspects of method reporting that I think have perhaps improved recently, as well as something in particular that I think is where there's room for development. And then I will briefly talk about what we as a field can gain from sort of embracing open methods more widely. So the core aim of comparative cognition is to learn about similarities and differences in cognition across species, and on the basis of these, make inferences about how these cognitive abilities evolve, and hopefully understand something about how about the selective processes driving this evolution. So it's an ambitious aim, and it's certainly not an easy task. And a few years ago, when several areas of psychology were sort of confronting the replication crisis and starting to identify issues with several of their findings, I wrote this article about potential challenges for comparative psychology and comparative cognition in particular in relation to our ability to do reliable research. And lots of other people, including Ben, were writing about this and thinking about this at the same time. So some key sort of challenges I identified were things like a lack of replication studies, insufficient sample sizes, a lack of within study species diversity, relatively few publications reporting null findings. And then this point that I'm going to pick up on is approaches to task design. So what we at this point in time saw a lot of in comparative cognition was, and that's, it's the nature of the groups we work with and the questions we want to ask. What we tend to see is lots of novelty in methods design, where a research group identifies a cognitive ability of interest. They design a novel task to get at that in their target species. They use that to test the species and find sort of evidence for or against that ability, typically without doing anything in terms of validating that task. And I completely, I'm completely hold my hands up to this. I'm someone who has worked like this. And so it goes on. Someone else interested in asking a similar question with a different species might take that task, but adapt it quite considerably through necessity to be appropriate for their target species. And then what happens is we try to make comparisons between species across studies, which raises a whole host of issues. And I think there's been a lot of progress since then, since when was that, 2021? So, and a lot of that is due to advances in open science as well as big team science. So I think many primates was up and running at this point. Many dogs was in the works, I think, but these things are in very early stages. But thanks to these projects, we now do have things like studies that replicate previous findings with really, really large samples and a lot of species diversity, thanks to things like increasing uptake of pre-registration and registered reports. We see more publications reporting null findings. But I think we are still facing this challenge of task design. I think that problem remains. And one of the key challenges for us, if we want to compare diverse species, is that, of course, there are species-specific non-cognitive factors that can confound test performance. And these include things like a species morphology, perceptual abilities, their motivation to engage with a task, the ecological relevance of a task to them. So just think, for example, of the trunk of an elephant, the beak of a hornbill, and the hand of a chimpanzee, how different those things are. What the sort of issue that this really raises is this issue of performance versus competence. So if we find that a species fails a particular task, are those negative results due to a lack of knowledge or a lack of competence, or are there performance limitations due to the methods not being appropriate? And we can make adjustments, of course, and that's what happens, but these can be at the expense of our ability to compare. And this challenge only increases with the increasing diversity of species being tested or being compared. So if we think about the many's projects and the sort of diversity of comparison in relation to taxonomy, with knowledge that the similarity decreases as we move from species through to kingdom. With many babies, we're comparing within a single species and even within a very narrow age range. With many dogs, we're comparing within a species, but there are lots of different breeds. So already there's a lot of variability here. Many goats is another example of comparing within a species, but where there are different breeds. And we have many primates where we're comparing different species within an order. So we're seeing increasing diversity. So comparing something like a huge gorilla with a teeny tiny tamarin. Many birds, where what we're doing now, comparing across several orders. Many fish across several classes. And then we have many manys, where we're trying to compare all of these plus potentially a bunch of other species. So, when you think about that, it literally is quite mind-blowing. This is a really challenging task, a great, fantastic project to be doing. But I mean, it really does bring home the challenge of species fair design for sure. So how do we go about trying to design fair methods that are appropriate for really diverse species? So one approach is to have identical methods as far as possible, so within reason, which of course gives you a high level of direct comparability, but this is likely to be at the expense of ecological relevance or motivational value. And it risks making it not a fair test. So you might have seen this cartoon before. So it's a kind of fairly tongue-in-cheek example, but I think illustrates the nature of the challenge we're facing and the issue if we really do try to standardize everything. We end up with something that's just not a fair test. And we come back to this issue of competence versus performance. So if we have negative results or null results, is it that an issue, is it that the species doesn't possess that ability, or is it that there's something about the methods that aren't appropriate for them? So just to give an example of where I think this type of approach has been used. So the primate cognition test battery, I'm sure many of you are familiar with, is a suite of cognitive tasks designed to test physical and social cognition skills in primates, initially used with great apes, including children, and subsequently adapted to be used with monkeys, and then later used with parrots. And the adjustments to the task, as I understand it, were really quite minimal. So the same battery of tasks was essentially used with birds as has been used with primates, with some size adjustments and things. And what the researchers found was that despite the fact that parrots had previously exhibited quite sophisticated cognitive abilities in lots of the domains from the primate cognition test battery, in the primate cognition test battery, they tended to perform poorly. So at chance or, typically at chance level. And so this led them to speculate that maybe these methods aren't appropriate because they were designed with primates in mind. They were designed to be ecologically relevant for primates. I was interested to read about what's been sort of termed a two-pronged approach. And I think Laurent has done some work which could perhaps fall into this category. So comparing fish and primate species on a decision-making task. So you have got some pretty different, morphologically different in a lot of ways, species there. So my understanding of this is that there was a task that had been used with fish to test their decision-making. And the approach that was initially taken was this kind of direct comparison approach, keep things as similar as possible. Let's test capuchin monkeys with this same task. And somewhat surprisingly, the primates didn't perform so well at this. And again, it was sort of speculated that this might be due to the ecological relevance of the cues. And so subsequently, so there was this identical methods version, and then subsequently the task was adapted to maximize the ecological relevance for each species. And in that version, the capuchins also performed well. So this seems like a really interesting approach and allows for exploration of the influence of these non-cognitive factors, things like cue type. I do think we need to be careful to some extent. We need to draw a line somewhere to make sure we're not just tweaking procedures or criteria until we find evidence of an ability of interest. And while this seems like a really nice approach with perhaps a small number of species or within smaller scale studies, it's trying to think about how an approach like that might work at the scale of a big team science, or in particular many where there are so many species involved, that could be a little more challenging. So I think what perhaps where we land is something like aiming for functional equivalence, which is where we're trying to balance the standardization of what we decide are the core components of a task, and then we allow other things to vary to meet species-specific requirements. And by doing this, we maintain some level of direct comparability, and we increase the likelihood that participants display a particular cognitive ability if they do indeed possess that. So we reduce the likelihood that competence is being masked by performance limitations. And at a quick glance, it seems like this might be the approach that some of the other Menis projects are taking. So the first many primate study looking at short-term memory, some core components were standardized. So there was a rectangular board with 3 identical opaque cups. And then lots of other things were allowed to vary, some of which may have more influence on performance than others, but quite a lot of things were allowed to vary here. And similarly, the first Many Birds project looking at neophobia, certain core aspects, core components of the task were standardized. So here it's the visual object properties that were relevant. So the same colors were used. There was something shiny in all of the objects, but they were adjusted significantly for body size if you think about the range of bird species. When it comes to reversal learning, so this is not an area of expertise for me at all, but I did enjoy sort of having a quick look at the comparative cognition literature on this, and think it seems a little more complex to me in terms of what the core components are and what would need standardizing versus what could vary. But in case anyone's not familiar with reversal learning, so it's generally taken as a measure of cognitive flexibility, and an individual first learns to associate one of 2 stimuli with a reward. And then once they reach some set learning criterion, the reinforcement contingency is switched so that the previously unrewarded stimulus becomes rewarded and vice versa. So in a typically used paradigm like the one depicted here, so we have a vervet monkey here lifting a pink cup. So they would first learn that a food item is hidden under a pink cup, not the blue cup. When they reach a learning criterion, that then switches. So now the food is under the blue cup. And the aim of Many's one, as most of you will know, I'm sure, is to develop and implement an innovative task to measure, describe, and that bit's hidden under my screen, and compare reversal learning abilities across taxa. So in terms of what types of paradigms are out there, there were several similar to this vervet monkey one, where food is hidden or covered by a cup or container or cover, and color is used as a cue. So in all of these, with primates, birds, fish even, they have to learn which cover or container a food item can be found under, and then that's reversed. Shape can also be used as a cue and has been, to look at, say this was a study with cichlids and bamboo sharks. Sometimes touchscreen paradigms are used, so the sort of format of presentation of stimuli varies. This is, again, a finding food under a cover, but now the cue is spatial rather than color. So it's the relevant cue is whether the food's on the left or the right. This is a kind of Skinner box type version with mesocarnivores, so raccoons, skunks, and coyotes, where they have to learn which of two buttons to press to get the apparatus to release a reward. So again, that's a spatial cue. And then there's the variety of sort of other paradigms with a detour task with a jumping spider, again, where color's the cue. There are visual cue studies with butterflies, studies with bees, where there was often a visual cue, but sometimes there was an olfactory cue. And there was a picture book study with toddlers. So lots of species tested, lots of methods used, but sort of typical of what we see in the field. Lots of studies with one or a few closely related species, each with sort of idiosyncratic methods. So I think, okay, I guess to try and sort of summarize those things that I took away from having a quick look at this literature. The stimuli or the type of cue was one thing that varies between studies. The response mode, so it might be turning over a cup, moving towards a stimulus, pressing a button, touching a screen, a feeding attempt. The sort of cue reward contingencies, so it feels a bit different to lift up a cup and have the food reward right there, to pressing a button and then after some delay, some food being dispensed. The outcome of making an incorrect choice was also different, so it might be no reward, like in the cup studies. In the touchscreen studies, it was typically a timeout, and there were some studies where it was an aversive stimulus. And then the learning criterion, it was, when is it that we can say a species has learned a reversal? So just to highlight a couple of examples of within species comparisons to sort of highlight the potential issues if too many things or if certain things are allowed to vary across species. So this is a study by Mark O'Hara and colleagues comparing the reversal learning of Kias in a paradigm where they had to turn over, so kind of like the cup paradigm, but with more distinctive 3D objects. So this sort of 3D fish and 3D duck covered over a food item, and they had to learn which one it was under. So they compared their performance on that with their performance on a touchscreen version, where they were actually seeing images of these things, but they had to learn and reverse on the touchscreen rather than on the with the physical objects. And they found this solid object version easier than on the touchscreen. And there was a similar example with comparing color cues and spatial cues within a very similar paradigm with hornbills. And I think the crucial thing to highlight here is that if cue type had been allowed to vary between species, then you could end up erroneously concluding that one species was better at reversal learning than the other. So I think it's going to be challenging, but it's going to be up to many as a group with expertise in the study of reversal learning and in the behavior and cognition of all these different species to establish what are the core components that need to be standardized and what can be allowed to vary. And I think it's challenging. Okay, so I'm going to move on from that now to just spend a few minutes talking about these other sort of prompts that we were given. So what's missing from methods reporting? I think transparency of methods has improved a lot in recent years. So pre-registration and registered reports mean that we can be clearer about things like inclusion, exclusion of participants or data points. There's less opportunity to make decisions about those things after the fact, and they're transparently reported. Open data and code means that we can reproduce analyses, and infrastructure like the open science framework means that we can share fuller methods. So sometimes journal articles are restricted in terms of how much you can report, and they have supplementary material, but then if you actually try and find that, it's not always, as a reader, it's not always easy. So if you can share links to materials on open science framework, that allows for fuller sharing of methods. One thing that I think is a potential area for development is the reporting of pilot studies. So, and not necessarily, well, definitely not unique to comparative cognition, but potentially particularly important for the field and for a project like this. So we pilot for all sorts of reasons, but it's typically we're pre-testing something about our study before we start data collection for real to see if things are working as we think they should. And within sort of single studies or single research groups, it can be really challenging to pilot, or you have to weigh up the value of it, because we're often working with really small numbers of participants. So I've run studies with 5 orangutans or 6 capuchins per condition. If I'd use some of those as pilot subjects, I'm reducing an already very, very small sample even further. But perhaps with, grouping together and pooling participants, it's more feasible within big team science projects. And it seems particularly crucial when developing tasks for really, really diverse species. A sort of related point is that I think it's really important to keep track of the methodological decisions that we make and how they were arrived at. I sometimes struggle to think back to decisions I've made and why I made them. But in a huge project with a huge network of members and people changing roles, I think that it's particularly important to be keeping track of methodological decisions. So in terms of what we see typically with pilot reporting, and this is based on some really recent work by the Pilot Reporting Task Force, which was established at a SIPSUM conference. It may well be that some of you are involved in this. I only learned about it recently. But they are working on improving the transparency of pilot reporting. So we pilot for lots of reasons, but piloting really can influence the methods we ultimately use and the decisions we make. However, in terms of how we report this, it's typically fairly vague. I myself have definitely written in papers based on pilot work, this decision was made, without providing any further detail anywhere else. We're encouraged to present a kind of polished version of our studies through the publication process, and we're hopefully getting better at not file drawing whole studies, but essentially we are currently file drawing this part of the research process, which is a really crucial part. And by transparently reporting our pilot studies, there's the potential for readers, reviewers to identify selection biases that might be harming generalizability. And this will help to evaluate research for readers, reviewers, the public. And also this type of information, I think, could be really, really valuable to other researchers. So, we all want to know, has someone tried this before? What might happen if I increase the number of trials? If this type of stuff is documented, it could be a really valuable resource. And this pilot reporting task force is working to develop templates for transparent reporting of pilot studies. But I think there's definitely room for innovation within comparative cognition and big team science. Finally, what are the benefits of embracing open methods and sort of working collectively? Well, there are practical benefits in terms of transparency and accessibility for members of these big projects and beyond. I think there are inclusivity benefits, particularly if we share data, be that, spreadsheets of numbers or even videos. I think it provides potential opportunities for those without access to research participants or for training of students. But sort of more fundamentally, I think it can lead to acceleration of progress with what we're trying to achieve as a field. And by combining open science practices with big team science, we can make better progress at addressing the questions of interest to comparative cognition. And we can do this better than if we work as sort of single labs or research groups. And finally, we can have more confidence in our findings and their reliability. So just to sum up, I do think progress has been made since I wrote that article, and that's thanks to the advent of things like big team science, advancing open science practices, and shout out to all the early career researchers who have really been, driving this forward and spearheading this stuff. There's no getting around it. Designing appropriate methods for diverse species is really, really challenging, and there's no silver bullet solution. One potential point of compromise is to aim for something like functional equivalence, but I think it's difficult to get a large group of people to agree on what that looks like. However, this group of people sort of stands the best chance of doing that, right? There's the relevant expertise in the cognitive ability of interest and in all of these different species. A potential area for development that I've identified is pilot study reporting and the transparency of that. And all of this is a lot of work, but I think ultimately it's going to enable us to answer questions beyond the scope of individual studies and have confidence in our findings. So thank you very much. Good luck. And I look forward to the discussion.
Thank you so much, Emma. That was a great talk and a lot of things for further discussion. I'm sure that everybody has questions and comments. So yeah, thank you so much.
Thanks.
That was great. And now we'll welcome Ben Farrar, who's going to give his perspective on open methods in comparative cognition research. So over to you, Ben.
Yeah, thank you very much, Nico. And yeah, thanks to Emma for that great talk. I think I agree with everything she said, and it's probably better said than my take on it. So yeah, we'll see. I'll share my screen and see if you get the slides. So hopefully you have my slide deck up now.
Perfect.
Amazing. So yeah, I'd just like to start off by saying thanks for being asked to do this. It's an honor, and it's great to see so many people out here. It's over 75 participants, so that's amazing. So kudos to the organizers putting things like this together. It takes a lot of effort, so well done. So yeah, I'm going to talk similarly on open methods in comparative cognition. I want to start by recognizing open science is this very, very broad term, and it's great to see many's and also a lot of the other many projects ticking off so many of the different foundation blocks of open science. We're probably going to be focusing on a more narrow sense in this meeting, given that we're talking about open methods and particularly designing species fair tests. But just wanted to put that out there. So like Emma, I got the brief of these 3 prompts. How do we build the best species fair comparisons that we can do, and how can open science help? What aspects of method reporting might be missing in comparative cognition publications, and what does success look like for the field, and what can we gain? I want to start at the end by saying something which I've believed for a long time now, is that the first thing we can get out of embracing open science, embracing big team science, is to understand the expected magnitude of between site variability in animal behavior across a range of tasks between species and within species. I don't think we understand that yet. And until we do understand that, a lot of the previous research we've done in the entire field is really, really hard to understand. And I think this is the most important question in our field. And I think it is answerable, and it is answerable through open science and through big team collaborations, along with other research methods as well. So I'm going to say this a few times in this talk. But for species fair comparisons, so as I was introduced, I've actually kind of moved out a bit of comparative cognition research at the moment into research in drugs and clinical trials. So I actually want to sort of take a different lens on this, because we face the same issues day to day in my job. And how do we make drug fair comparisons? And there's been a lot of research into this. So how do you say, okay, well, you've got drug A and drug B, how do they compare against each other? And we're trying to do species A and species B, how do they compare against each other? So there's a huge framework that we could lean on in the field of clinical trials and in drugs. You're probably screaming at me, well, you make drug fair comparisons by doing direct comparisons between them in randomized controlled trials. And you have drug A and drug B, and you randomize participants to each arm. And if a few assumptions hold, like the randomization being fair, sufficient blinding, then you can get a really good estimate of the relative effect between drug A and drug B. And that's true. And you're probably also saying, but Ben, we can't randomize, we don't have individuals to randomize to species A and species B, so this doesn't work. And that's true. So we can't have the same species in the same trial and randomize them. So scenario one, I've termed it, of a randomized control, the direct treatment comparison doesn't work. Another scenario that happens a lot is you have two different sets of direct treatment comparisons. So you have two different, so you have your first clinical trial, you have species or drug compared to placebo. And then you have a second randomized control trial. So you have your drug A compared to placebo and your drug B compared to placebo. Now we might be thinking, okay, it's getting a little bit more similar to what we face. You have different sites and different trials going on. Again, here, though, they are still randomized control trials. So if you have the assumptions of fair randomization, appropriate blinding, et cetera, you can do something called an anchored indirect treatment comparison. So if you know drug A is 5 points better than placebo, and you know drug B is 3 points better than placebo, then you can make the inference that drug A is 2 points better than drug B. This layers in another round of assumptions. So it assumes there's balanced effect modifiers between studies. So this is getting something similar to what we're hitting at, right? So this only holds if there's nothing that in the first comparison, so maybe the first comparison's in a bunch of people that are over 70 and this drug really works really well in the old people. And then in the other comparison, they're all under 40, so the drug doesn't work. So it doesn't hold if you have imbalanced effect modifiers, but if you can demonstrate you have balanced effects modifiers or essentially functional equivalence, then this sort of framework works. So this could be something we could see if we can anchor into. There's a third scenario where you have unanchored indirect treatment comparisons. So you have drug A and you run a study on drug A and there's no placebo control for it. So you have a group of people being studied with drug A in location Y and a group of people being studied on drug B in location X. Through this, you can make a comparison between them just by seeing the outcomes of drug A in location Y versus drug B in location X, just take them away from each other. Now we lose all the assumptions of the randomized control trial because they're not being randomized. There's no blinding, et cetera. We still have this assumption of balanced effect modifiers between studies, but we have an additional assumption layered in here as well, is that there's no imbalances in prognostic factors between studies. So in the drug example, if you're looking at survival and drug A, you test on a load of people who are 70 years old and drug B on a load of people who are 40 years old, it's no surprise that the people who are 40 are going to live longer. So that's a big confounding in your design, and that's an imbalance in prognostic factors, independent of whether the drug actually works better or worse in the age. Age itself is a prognostic factor that affects performance. So my question to you to think about for about 10 seconds before I go to my next slide is which scenario do we find ourselves in comparative cognition, or do none of them apply? Kind of hinted at, okay, well, we're not randomizing participants at all in comparative cognition. You might say we could try and force it into an anchored indirect comparison by making our tasks the same, but this still relies on the benefit of randomization. So in comparative cognition, I think we face a very similar situation to unanchored indirect treatment comparisons. And there is a formal framework for understanding bias in this and estimating bias and seeing how we can work with this. And I think we could, as a field, learn quite a lot from it. And it's good because a lot of people have done a lot of the thinking, and we get analogous things coming out, we're just using different terms. But this is an established field from many years. So we want to zoom in on these assumptions. And essentially, if you achieve balanced effect modification between studies and no imbalances in prognostic factors, then you have functional equivalence. That is what functional equivalence is in clinical trial speak. However, it's very, very hard to do this. And I want to pull out a couple of commentaries on unanchored indirect treatment comparisons from the medical literature, just to kind of exemplify what a challenge we actually face as a community. So this is from the EU that have just released some new guidance on this. Naive comparisons, that's unanchored indirect treatment comparisons between treatment arms in such situations are prone to bias and confounding and should not be performed. And this is echoes everywhere in the literature. So this is another paper, but you can find thousands, or maybe not thousands, hundreds of examples of this. Naive indirect comparisons are in most instances inappropriate and should only be used for exploratory purposes and when no other options are possible. As a field, though, we do sit in the category of when no other options are possible. So we do have to do these. So we have to make the best of what we can do with them. In clinical trials, they try to overcome this by using statistical adjustment or matching patients to overcome this. So there are ways to combat it. And this relies on the assumption that after you've done your statistical adjustment and matching, that no residual confounding remains. Now, that's usually not true. And that's what we'll face as well. But they have a tool to address this called quantitative bias analysis. So what's the size of the residual confounding that would be required to fully explain away effects? So say test species A and species B, you get a difference of say 20 points. Is it realistic that residual confounding would lead to a 20-point difference? Is it realistic that the imbalances in prognostic factors or effect modifiers would genuinely lead to such a big difference? And you can say, okay, well, based on our knowledge of the effect modifiers and prognostic factors, that's unrealistic. So we're pretty confident this effect exists and it should exist in this range of effects. The trouble we face, and what I think many can contribute to, is understanding what the prognostic factors and effect modifiers are and understanding how big this degree of bias could be that comes from them. So on the flip side, in comparative cognition, we design tasks to be species fair, functionally equivalent, to reduce this effect modification. So this strong assumption of no residual confounding remains. And I think in comparative cognition, functional equivalence hits the balanced effect modifiers somewhat, but we don't really talk about imbalances in prognostic factors. So this would be so an effect modifier might be the size of a cylinder if you're doing cylinder tasks. So you would want a bigger cylinder for an elephant than you would for a mouse, and that would modify the effect that they would have or modify their performance. But you could have prognostic factors as well that are imbalanced. So this could be, say, the age of the animal. So you're testing, a bunch of 10-year-old capuchin monkeys. I don't really have a frame of reference there. Versus 40-year-old parrots, maybe that's going to affect something because they differ in prognostic factors, not necessarily because the task could be functionally equivalent and it could match it all, but there could still be differences between these samples, which we will face, that are prognostic factors. So it's that extra level, and that's that extra thing we have to be careful about when we're doing these unanchored indirect species comparisons, when we can't rely on randomization, which we can't, and we know we can't, it's impossible. We have these two layers of things. So functionally equivalent tasks, and then also trying to, well, we can't balance the prognostic factors because that we can't do it, but we can try and understand them and understand the degree of confounding that comes from them. Where does open science fit in? Well, I genuinely believe that we're tackling one of the more difficult problems conceptually in science, and we have a very small amount of data relative to other fields to do this. So the only option really is to have good sharing of data, good sharing of methods, and also what's really great about initiatives like this is you're opening access of species to different individuals, essentially. We need data, and but we also need all the sort of scaffolding around this. And I think Emma talks about that with sort of pilot tests and things like that. That's all going to be sort of effect modifiers. How much piloted have they been on these things? So we need more sharing and more data. And we can start to build up an understanding of how much this behavior varies and what the difference is, what the magnitudes associated with different factors could be. And I think that's the success factor. And I would say I don't think it is all doom and gloom. There are things that we have going for us. So we know species are very, very different, right? Maybe drugs aren't so different. So the effect sizes that we might see between species might genuinely be quite large on a lot of tasks. So then our job is to say, okay, well, the magnitude of unmeasured confounding is smaller than what this effect size we see. So we could see large effect sizes, we might not, but we need data to be able to quantify this, and we need to see the stability in effect sizes between groups, between sites, to start pulling these things out. Yeah, so as I said, again, I think the success factor is to understand the expected magnitude of between site variability in behavior, understand these effect modifiers, prognostic factors, and what sort of effect sizes we're going to see from animals and between species. And I think there's a unique role that the sites that have multiple instances of the same species can play here. So many dogs, many goats. I mean, you're going to have it a lot of the time, but that's really, really key, I think, as a starting point to understand within the same species, how much is it bouncing around before we move to within different species. But we can do it all at the same time. There's no need to say we have to do something first, but there should be good anchor points. So as Emma, I spent most of my time on species fair comparisons. I'm going to touch on open methods now. This is a bit more scattered. So what's missing from method sharing? And I got this example from, that's definitely not 2025, sorry for that, so it was 2021. A bit of research that I was involved in with Piero Modio. And we often see diagrams like this in publications, and I think they're really good, they're really pretty. You get a rough understanding of what the task design is. Sometimes you see them with the exact dimensions marked out. If you go back, this was 25 centimeters, that was 30 centimeters. Really now in 2025, we can provide this, but we can provide so much more. Because when I go into papers and animal cognition, I often see a picture and I say, okay, I've got a rough understanding, but I don't really know what's going on in the task. So if we want proper open materials and proper transparency, I think the way forward is high quality sharing of videos, of task setups. You can learn more from a video about a task than you can from reading the methods. Or well, in tandem, you get a much fuller picture if you have the videos as well. That's not to say that every video is a great video to show. So I think we want videos of the full test, including pre-test procedures, because this is going to be a whole host of, from having worked with animals, you can't just start the video from when the animal starts interacting with the test, right? Some animals are really hard to recruit, some really don't want to be there, some really do want to be there. That's important information that if we want to actually understand the meaningfulness of the data you're getting, especially if you're hitting floor effects and ceiling effects in some participants, the task doesn't start when the animal interacts with it, it starts way before that. And I think this is kind of what Emma was getting at as well with the piloting somewhat. The other issue we have with sharing videos is, of course, people are going to share their best videos, and that's not going to give you a picture of what actually happens in the test. And there's a difficulty here because you can't, I'm not saying you've got to upload hours and hours of footage of every single participant. That's not feasible. It's not feasible from a data storage and time perspective. But as a field, we've got to look, okay, well, how this is a wonderful resource that we can have quite low cost and we can share it between things. How best can we do this in a way that's not biased? And I don't have the answer, but the answer is out there somewhere. We can do it better than what we're doing. And I actually had this as a nice example here from Penny and Hare, 2018, because they've been sharing videos for a long time. But that's my sort of open research 2.0. I want more video sharing. I won't touch upon these other 2 points, but they're also really, really important, is blinding, how we perform it and how we report it. I think it's underreported, and it can have a massive influence on animal performance. So we don't do it enough, and we don't report it well enough when we do report it. And I think Alize might be on the call, but she's writing a bit of a paper at the moment, or from some research that we did, and there might be other collaborators on here that characterize how people report blinding inter-observer reliability, and it is patchy or has been patchy. Inter-observer reliability, so you have people coding videos and you say, okay, well, there's 85% agreement or whatever. To understand what that means, you have to understand how they trained on these things. If two people train together with a sample of videos and you overfit yourselves, and then you're not independent coders, right? So you could use networks like this to get truly blind independent observer reliability coding. So people should be trained enough to be able to understand what's happening, but not overtrained on it. I think overtraining of inter-observers is probably a big issue. And also understanding where the final data set comes from. So you have 85% agreement, but what happened in that 15%. That's another area of flexibility in there. So that's it for current open methods. Aspirational would be to start characterizing. We have animals that we have repeat performances of. I would love to see, if you've got young animals, really good recording of their baseline characteristics. So in clinical trials on the right-hand side, as you see, you always have this big table one of, okay, well, this is the age, this is the participant, this is their, you know everything about the participants. And this comes back down to these effect modifiers and prognostic factors. When you go in a comparative cognition paper, you might get the age of the animals, maybe, and maybe a bit of wording on how experienced they are with the task, but not enough information if we want to have a full go at prognostic factors. So if you're starting a new lab, you could carefully record data throughout, and people do that, but things like age, number of tasks you performed, you may even be able to get characteristics linked between studies, right? So you could be reading an article now and you could say, okay, well, these animals did the cylinder task 2 years ago and this was their score. And then all of a sudden some other lab will say, okay, I've got this data. And then you can start saying, okay, well, they've performed that well on that task and that well on that task. And you start to build up a bigger picture. And this is the type of stuff that you would need if you wanted to go forward into statistical ways of adjusting between species comparisons between sites. I put it as aspirational because it probably doesn't work. But in an ideal world, it's the type of data that you would have. So yeah, that's it from me. I think species fair comparisons, it's genuinely a really hard scientific question. There are frameworks we can use to understand the problem a bit more. I think Emma's given a really good example of building up with lots of nice examples from our literature about how we can go about this. I think we're all in agreement that sharing data, methods, and access to species is crucial to answer this question. Methods, I think unselected videos or some effective way of sharing videos of task performance is brilliant, both in terms of with the publication, but when you're designing collaborations like the many-manys, sharing them between labs ahead of time and being happy to share videos that might not be optimal with each other so you can build these learnings from it. Reporting of blinding, inter-observer reliability, really nailing these down as a field would be great. And then aspirationally building up a way to building as big a profile of the animals that are doing the tasks as we can do. And then with all of that, we should be able to start making some steps towards understanding the expected magnitude of between site variability in animal behavior across a range of tasks and species. So yeah, thank you very much.
Thank you so much, Ben. That was fascinating. A lot of things to discuss. From the two presentations, I've got a page full of notes, and I'm sure other people have questions and comments too. So Laura will moderate the Q&A now.
Yes, I'm happy to take over. Thank you so much, Ben and Emma. Those were very, super great talks and very inspiring.